Welcome to EduRev.
Welcome students. I'm Clarice Pereira Colasso and I have been training NEET medical aspirants in botany and zoology as well as NCERT based board exams for the past 8 years. In the previous video lectures we have completed pre-fertilization as well as fertilization structures and events in case of flowering plants, and we have also completed endosperm formation as well as embryogenesis, that is a post-fertilization event in case of flowering plants. Today will be the last video lecture on sexual reproduction in flowering plants, and this will include the following subtopics. The first, the structure of seeds and what is their importance. Okay, so what are the different types of seeds? The next is what are the different types of fruits and what is the concept of apomixis and polyembryony. So first of all, the structure of seeds. Now actually speaking, seeds can be divided into three distinct parts. The first is how many cotyledons do they contain, single cotyledon or double cotyledon. For example, monocot has one cotyledon, dicot has the presence of two cotyledons. The next is you have an embryonal axis. So the embryonal axis helps you to divide the embryo. And the third is the presence of seed coats, which are protective structures of the seed. Now to start off with seeds, let's start with the dicot seed. So here you can see the presence of two cotyledons indicating dicot, okay, and in the center you can very clearly visualize the red line that is drawn over there as the embryonal axis. So the dicot seed clearly has these two parts along with the presence of the seed coat on the outside. Now generally speaking, seed coats are two in number. We will very soon understand why two in number. Now, if we look at the dicot seed, now this is a cross section of the dicot seed, you can see that the endosperm reserve is almost negligible. So very little endosperm reserve present over there. Now remember, dicots have two cotyledons, okay? And now the very fact that you do not have a proper endospermic reserve, we can say that you're a non-albuminous or you're a non-endospermic seed. Now the question is, why would a dicot seed prefer to just have its cotyledons and not the endosperm? Why not have the endosperm as well? Now it is found that these particular cotyledons, since they are two in number, the whole concept is since they are two in number, are able to provide more than enough nutrition to the developing embryo, or rather it helps in the development of that particular embryo into the seedling. So it is more than enough nutrition for germination. So the two cotyledons are more than enough. You do not require an extra endospermic reserve. So the examples here are very important for the NEET exam. That is the case of pea and in case of groundnut. Now coming to monocot seeds. When you look at monocot seeds, very clearly you can visualize that the endospermic reserve is way bigger, way greater as compared to the previous case, okay, as compared to dicots. Now the question is, why is the endospermic reserve so important in case of monocots? Remember, they have just one cotyledon. So that means as this particular embryo is germinating so as to form the radicle and the plumen, in other words the seedling, it requires a lot of nutrition, and that one cotyledon is not sufficient nutrition. Therefore, it must rely on the endospermic reserve. Therefore, these seeds are also going to be called as albuminous seeds. Very, very important. Now, the outermost layer of the endosperm here is going to be called as the alluron layer. Moving on, the examples again are important. So you have the example of castor, maize and wheat. Now to understand the structure of the seed, we must understand or we must revise through the structure of the ovule, because the various parts of the ovule are then converted into various parts of the seed. For example, the funiculus was meant to be an attachment point between the ovule and the placenta of the ovary. However, once fertilization takes place, that same funiculus will hold the seed inside the fruit. So it will give the seed a position in the fruit. The next, we note that the ovule has the presence of two protective coverings called as the integuments. Now, these particular integuments after fertilization will be converted into the seed coat. So if you have one integument, you will have one seed coat. If you've got two integuments, you will have the presence of two seed coats. The next part is the egg cell. Now the egg cell was obviously fertilized with the help of the male gamete, so therefore the egg will result in the formation of the zygote. But let's come back to the integuments. The integuments seem to be They're not a complete layer. They seem to have a small opening in the center called as the micropyle. Now the initial purpose of the micropyle in the ovule was to allow the entry of the pollen tube, and then it would enter into the filiform apparatus of the synergid. However, after fertilization, this same micropyle remains as an opening in the seed. Now, why is it required as an opening in the seed? Why does the seed need an opening? Now, the purpose of this particular opening is so that oxygen can enter into the embryo, or rather the embryo can be fed with oxygen, and the cotyledons can also be fed with water. Now, why does the cotyledon need water? Remember, the cotyledon has the presence of food reserve. However, these food reserves are in a concentrated form. If the embryo must utilize these food reserves in order to germinate, these food reserves must be diluted with the help of water. So therefore, the micropyle opening serves as an opening through which water and oxygen can enter. So in other words, it will facilitate germination eventually. The last part that we need to focus on is the nucellus. Now, actually speaking, the nucellus of the ovule after fertilization should degenerate. However, in certain cases, you will find that the nucellus does persist. Now, if it persists, for example, in case of pepper, black pepper balls, okay? So in case of pepper, you will find that it persists. Now, if it persists, it's going to be called as the perispore. Again, a term very, very important for the NEET exam. Now, coming to fruits, we need to know what are the different types of fruits. Now, fruits can be classified in different ways, but basically a fruit is of two types. One can be called as a true fruit and the other can be called as a false fruit. Now, we'll take two different types of fruits in order to understand what are true fruits and what are false fruits. Now, on the left hand side, we have the presence of a mango, which is a true fruit. On the right hand side, you have the presence of the apple, which is a false fruit. Now, what makes one true and the other one false? Fruit formation is actually speaking directly from the ovary, which means if the ovary swells to give you a fruit, so fruitification takes place from there, that is going to be called as a true fruit. Now, in case of the mango, you will find that the ovary actually gives rise to the fruit. Therefore, it is going to be called as a true fruit. However, if you look at the apple, okay, the apple has the seeds in between and there seems to be a line structure all on the outside. Now, the part of the apple that we actually consume is not really the ovary. Rather, we consume the swollen thalamus. The thalamus is the base of the flower. Okay, so we consume that particular part. We consume the stalk, that particular part of the flower, which eventually has become the fruit, which is actually the thalamus. Okay, so we consume the false fruit. Now, if we really had to consume the true fruit, we would be consuming the central core. And generally speaking, when we cut and eat an apple, we throw out the core of the apple. Okay, so the true fruit is actually the interior portion, which is not really consumed in case of humans. Okay, so the difference between a true fruit and a false fruit. Now, let's look at what are the different parts of a true fruit. So if you look at a true fruit, first of all, if you look at the mango, you have a skin on the outside. So the skin is going to be called as the epicarp. Okay. The second part, which is the fleshy portion over here, is going to be called as the mesocarp. So this is the part that we enjoy eating. And finally, you have something called as the endocarp. Now, these are the three external layers. Okay. But what is the endocarp? Now, if you look at the seed of a mango, it has kind of a hairy outer covering. Now, that outer covering does not belong to the seed. That hairy outer covering, okay, actually is part of the outer wall of the fruit. So that is going to be called as the endocarp. It is a little hard in nature. So these three layers when taken together is actually called as the pericarp. So this term pericarp is absolutely essential for us. Now coming to something called as parthenocarpic fruits. If you listen to this term parthenocarpic, it should remind you of parthenogenesis. So parthenogenesis is a terminology that we have studied in the first chapter, that is reproduction in organisms. What is parthenogenesis? It is the formation of an offspring without actual fertilization, and we gave an example of honeybees in which the drone is formed without fertilization. So if we understand what is parthenogenesis, it obviously implies that parthenocarpic fruits is fruitification, so the formation of a fruit without actual fertilization. So you have the swelling of a fruit without actual fertilization, for example in case of banana. Okay, now also some varieties, some hybrid varieties of grapes can also be called as parthenocarpic. They don't have seeds on the inside. Okay, now how does one come up with such parthenocarpic fruits? Parthenogenesis or parthenocarpic fruits can arise with the help of hormonal treatment. But remember, since there is no fertilization, automatically we can understand that we arise with seedless fruits. Now this is advantageous for us, especially when we're talking about horticulture and we want to sell fruits. Now, coming to the advantages of seeds, why would a plant choose to provide or to make seeds? Now, the first advantage of seed is that it's a dependable process. Now, what do we mean by dependable? If you look at some of the pre-fertilization processes, events that took place, like pollination, okay, all of these were chance occurrence. But the moment you have the presence of pollination and you have fertilization, for sure you will get a seed. So it's a dependable process. Now, the most important purpose of a seed is it is good for dispersal. So you can make use of different agents that can help you to disperse. So plants have used this as their adaptive strategy to transmit or to disperse their seeds to a variety of areas. For example, if a bird is eating a fruit, as the bird flies, it takes the fruit from one place to another. The seeds would fall onto the ground of a second place, so you would have germination in a whole new territory. So it is an adaptive strategy on part of the plants. The next, seeds already have the presence of nourishment for their young ones, implying the presence of the cotyledons or the presence of the albuminous reserve, the endosperm reserve that could be there in monocot. These are already kept, okay, they're already there, they're already ready, and the embryo that's developing into the new plantlet can make use of these particular reserves. So, nourishment for the new seedling is already provided. The next, there's protection for the embryo. Now, how do we say the embryo is protected? Remember, the embryo is basically forming from the zygote, which is forming from the egg cell. And the ovule has the presence of the integuments, which are converted into seed coats. So those seed coats are protective in nature with respect to the embryo that's on the inside. Now remember, seeds contain the embryo. Embryos are generally formed by fertilization, so you are more or less sure that you could have probably variations, especially if you have cross pollination. Okay, so variations can be induced. Now the last advantage is not really an advantage to a plant, but it's an advantage to human beings. Since we have the concept of seeds, this is the basis of agriculture. Okay, so you can sow seeds and seeds will give rise to your crops. So this is the basis of agriculture. Now, we have already studied something called as viability of pollen grain. Likewise, there's also something called as viability of seeds. So seeds can be viable for months to years, again, depending upon the temperature, depending upon the humidity, okay, the type of soil given to it. So a whole lot of things that determine the viability of a seed. However, there are two seeds of importance to us. Now, one of them is Lupinus articus. Now, this particular seed was found in the Arctic tundra. It is around 1,000 years old, and they found that this particular seed was so well preserved that even after, 10,000 years old. So even after 10,000 years, when they sowed this particular seed, they found that it germinated into a plant. So you can understand that the viability of seeds can range to a huge amount, many number of years. And the second example that you need to remember for your NEET exam is Phoenix dactylifera. Now this particular seed is around 2000 years old, was also able to germinate, and this particular seed was found when they were excavating King Herod's palace around the Dead Sea area. So that's in Israel. Now coming to the last concept of this chapter, apomixis and polyembryony. Now, I'm quite sure all of you have seen citrus fruit. For example, you have lemon, lime, you have oranges, or you have sweet limes. So you have different types of citrus fruit. Now, when you look at the citrus fruit and you take each segment of the citrus fruit, each segment represents a carpel. In other words, each segment represents the presence of one particular part of the ovule, okay, or rather one particular ovary as such. However, inside it you will find huge number of seeds. Now the question is, how is it that so many seeds are arising in that particular region? Now, if you look at the presence of the embryo sac actually has the presence of the egg cell alone, which should give rise to one seed when it matures and when it's fertilized. However, remember, some of the nucellus cells that are present on the outside are still nutritive, maybe not as nutritive as the myocyte that gave rise to the embryo sac, but they're quite nutritive. So some of them can also undergo seed formation. However, if they do undergo seed formation, we must understand that these seeds were formed without fertilization. Therefore, what is apomixis? Apomixis is the formation of seed without fertilization. Now, obviously, if you're forming a seed without fertilization, you're forming something that can germinate without fertilization, it is a type of reproduction. So this is an asexual seed. It is a form of asexual reproduction rather, not sexual. Okay, and they generally develop from the nucellar cells. Generally from nucellar cells. Now you must understand these particular apomictic seeds will not have any variations in them. So in that single segment, you will find just one seed that has the presence of variations, but any other seed present there will be devoid of variation. In other words, they will be asexual apomictic seeds. Now, since such a concept is possible, we can obviously understand that many embryos are going to arise. So therefore, we can say polyembryony is the resultant concept from apomixis. So I thank you students for attending today's video lecture. I hope you have understood the entire chapter of sexual reproduction in flowering plants. I invite you to go through EduRev Infinity, where you can come across a whole lot of other videos, test papers and lectures, as well as notes prepared by NEET experts. Thank you.